Bismillahirrahmanirrahim. We're going to discuss March 24, 9700 paper double 2, which is the A level, AS level paper. Starting with question number 1, figure 1.1 is a diagram representing part of the phospholipid bilayer of a cell surface membrane. Look at it very carefully when you're reading this question. You can see the labeling. There's cholesterol, which is this part. Let's got this part. So this is the cholesterol. Then you've got the cholesterol here as well. You've got the cholesterol here as well. Then you've labeled fatty acid tails. So these are the hydrophobic part. These are the fatty acid tails. Then you can see the phosphate heads. This is the phosphate heads which they have shown you. Now look at it carefully and then read the question and then come back to the diagram again. Identify the part of a phospholipid molecule labeled A. Labeled A. What is this? So phosphate, then attached, and then there's the two fatty acid tails. So there's nothing very tricky, but of course you have to realize that this part is what? So it is glycerol. If you remember the structure of this fatty acid, there's glycerol and then there are three fatty acid chains. That is a triglyceride. But if you remove one of this and then you add a phosphate to this carbon here, this becomes a phospholipid molecule. So what was this? This was glycerol. I'm sure everybody would have come up with interesting answers to this. But it was glycerol. If you had looked at it carefully and realized what it was, I'm sure you could have fixed it. Is that the two fatty acid tails were attached to this thing. And so what was this thing to which was attached the fatty acid tails? Then coming to the part 2 of the question, cholesterol is an important lipid component of many cell surface membranes. Figure 1.2 shows the structure of a cholesterol molecule. So you can see this is the cholesterol molecule which they're showing you. Using the information in figure 1.2, explain the orientation positioning of the cholesterol molecule in the phospholipid. Now this is the hydropolar part. This was the polar part. They've labeled this polar hydroxyl group. And then this is the non-polar part. So you know the phosphate and the fatty acid tails. So the phosphate is the polar part. So this would be in the polar part. And this non-polar would be here in the fatty acid tails. So this non-polar part would be here. And this polar part, this part, would be here in between the phosphate heads. So the hydroxyl or the polar group interacts with the phosphate heads, and the non-polar part will be in the region of the fatty acid tails. The non-polar part would be in the fatty acid tail. So this is the non-polar part. So, I mean, yes, I used a green marker, so let me use the green. So this would be the non-polar part in between the fatty acid tails, which gives the non-polar part of the cell membrane. So writing it, the polar group interacts with the phosphate heads, or you could have said that uh, hydroxyl group faces the aqueous environment, or there are many words we could have worded it, but the simplest ways are these. Non-polar would be in the fatty acid tails, as the fatty acid tails, and this non-polar, they're both non-polar. But of course, you only had 1 mark, so any one of these would have got you the 1 mark. Then it says state one role of cholesterol in the phospholipid bilayer. Now this is very simple. Maintains fluidity, regulates fluidity, maintains or regulates the stability of the membrane. Either you said fluidity, or you said stability of the membrane, or you said prevents the entry of polar substances or ions. You could have said without the cholesterol, membranes would easily rupture. And of course, the reduces the lateral movement of the phospholipids. So lots of answers which you could have given me, in which you could have just given me the role of cholesterol. But of course, I find it the easiest is regulates fluidity, regulates stability of the membrane. I find these the best ones which I would have written. So you could have said any of them. There were a number of points which are available to you, but there's only 1 mark for it. Then coming to part B1, explain why sodium are ions, polar, charged, cannot pass across the phospholipid bilayer by simple diffusion. Sodium ions are positively charged, so they are repelled by the hydrophobic tails. Sodium ions are positively charged, so they are repelled by the hydrophobic tails, or you can say the non-polar core or the non-polar tails, or you can say the hydrophobic part of the cell membrane. Sodium ions are charged and so repelled by the hydrophobic tails, or the non-polar tails, you could have said that as well. Now coming to question number part 2, it says ions and some molecules move across cell surface membranes by facilitated diffusion and active transport. Compare facilitated diffusion and active transport by stating one way in which they are similar and two ways in which facilitated diffusion is different from active transport. So please remember to talk about facilitated diffusion first, because they have talked about it first. How is facilitated diffusion different from active transport? So remember to always look at the question and then answer accordingly. So similarities are three. Both occur through a membrane protein, or you can say a transport protein. Both can be specific to the molecule and ions passing through, or the molecule or the ion passing through. And both involve a conformational change of the carrier protein. Conformational change means there's a change in the shape of the protein in active transport and in facilitated diffusion. So number one, both occur through a membrane, a transport protein. Number two, both are specific. If I say sodium ions for facilitated diffusion, then that will only transport sodium ions from a higher concentration to a lower concentration. And then both involve a conformational change. That means there's a change in the shape. Now, this word conformational is a difficult English word, and I'm sure some of you would not have even used this word or know this word. So both involve a conformational change of the carrier protein. Now, differences, there are many, there are three which I'm going to talk about. Facilitated diffusion, substances transported down the concentration gradient. In active transport, it is against the concentration gradient. Facilitated diffusion is a passive process, does not require ATP. It's a passive process and does not require ATP. Active transport definitely requires ATP. And in facilitated diffusion involves channel and carrier proteins, but active transport does not involve channel protein, only involves carrier protein. Now, these are wordings that they use and which they expect you to know. So please know these differences. Similarities, I've talked about. Differences, very easy. Facilitated diffusion is high to low. Active transport is low to high. Facilitated diffusion does not require ATP. Active transport does require ATP. So very simple changes, simple similarities and simple differences. Now, as I've just given some of the brief of them, I haven't given all the points. So similarity is specific to the molecule, both are specific. Facilitated diffusion is for anything, it's only going to be for that sodium ion or for potassium ion or for calcium ions. And both of them involve a transport protein. Differences, facilitated diffusion is down the gradient, active transport is against the concentration gradient. Always talk about the one which is mentioned first in the question. And another difference is does not require ATP and requires ATP. Now coming to part 3 of the question, prostaglandins are small lipids produced in many tissues in the body. One role of prostaglandins is to cause inflammation at the site of an injury or infection. Inflammation is the normal first response of the immune system to injury or infection. Cyclooxygenase, COX, is an enzyme that catalyzes. So COX is an enzyme, so always remember it must have a specific active site. It catalyzes one of the steps in the reaction, and this is the reaction step. So that means arachidonic acid would be its substrate, and it converts that into prostaglandin. So this arachidonic acid will fit in here. So this would be arachidonic acid will fit in here, something which will be complementary. So this would be the arachidonic acid. So COX is an enzyme that catalyzes one of the steps in the reaction pathway for the formation of prostaglandins from phospholipids. So phospholipid to arachidonic acid and then to prostaglandin. The reaction pathway occurs in the SCR, the smooth endoplasmic reticulum of the cells. Remember the SCR? See, this is the nucleus, and then you have the RER, and then you have the SCR, which are these channels like tubes like. Part of the reaction pathway is shown in figure 1.3. Suggest an advantage of the reaction pathway occurring in the SCR, the smooth endoplasmic reticulum of a cell, rather than the cytoplasm. Basically, it is compartmentalization. You see, we don't want the things to come in contact with the cytoplasm. So what you have to understand is SCR can provide the substrate, or you can say prostaglandins are lipids and SCR is involved in lipid transport, or you could say prostaglandins can be transported from SCR or stored in the SCR. And the important point, which I always feel is a very relevant point, is compartmentalization. That is separated from the reactions in the cytoplasm. That's why you see mitochondria have a membrane, membranes around it, and chloroplast have membranes around it, so that the reactions taking place in the mitochondria and the chloroplast do not come in contact with the reactions taking place in the cytoplasm of any cell. The cytoplasm has ribosomes, has free ribosomes, so there are a lot of reactions taking place. Intracellular enzymes being made on the free ribosomes, extracellular made on the fixed ribosomes. So this compartmentalization is a very important topic, which of course is addressed in many, many questions. So you could have said any of those. Sometimes inflammation can have side effects such as pain. Aspirin is a drug that can be used to reduce these side effects. Aspirin reduces the catalytic activity of the COX enzyme. So it reduces the catalytic activity of this COX enzyme. Now, by modifying the R groups of one of the amino acids. You see, enzymes or any enzyme, COX enzyme or any enzyme is made up of a number of amino acids. So if it modifies the R group of one of the amino acids, what is going to happen? So just modifying the R group of an amino acid in the COX enzyme can reduce the catalytic activity of the enzyme. Very simple. You see, it could affect the bonding, the ionic bonding or the hydrogen bonding or the hydrophobic interactions with other R groups. Now, this would result in changing the shape of the active site. So this now would have an active site like this. So now if it has an active site, changes the shape of the active site. So active site is no longer complementary to the substrate or to arachidonic acid. So it'll affect the activation energy not being reduced. So enzyme substrate complex is not going to be formed. So naturally it's going to reduce the rate. So that's a very simple question, that why would why would the rate of any enzyme action be reduced? Because it will affect the shape of the active site. So the fact that they're saying that they're asking you in the question is they've showed you that by modifying the R group of one of the amino acids. So R groups decide the bonding. If there is sulfur on two amino acids, then forms a disulfide bond. Then different charges, hydrogen bond, ionic bond. So it will affect these bondings. If it affects the bonding, then the change is the shape of the active site. So the active site is no longer complementary to the substrate or arachidonic acid as in this question. So no enzyme substrate complexes will not be formed. So, I mean, wording it effect will affect the ionic bonding or the hydrogen bonding, or even you could have talked of the hydrophobic interactions with the other R groups. So it will change the shape of the active site. Active site will no longer be complementary to the substrate, and enzyme substrate complex will not be formed. So any three of these, I'm not saying all these four were needed, any three of these and it'd be fine. Then coming to the last part of the question, the long question, 13 marks question. Prostaglandins are an example of cell signaling molecules. Outline the process of cell signaling that leads to a response by the cells involved in inflammation. Very simple points which are in every question is the same thing. Prostaglandins are, they've told you prostaglandins are cell signaling molecules. So prostaglandins are secreted or released by the cells, or you can say are transported to the target cells. Then prostaglandins will bind to receptors on the target cell surface membrane. And then, of course, they will result in an enzyme cascade, or you could talk the signal transduction. And it was only for 2 marks. So you could have easily got your 2 marks by just saying that the prostaglandins are transported to the target cells and then they will bind to receptors on the target cell surface membrane. Target cell will have specific receptors on it, which are glycoproteins or glycolipids, which will be complementary to the shape of the secreting molecule. So the target cells will have receptors on it. That's how they will know that there's where they have got to bind. So prostaglandins transported to target cells, bind to receptors on target cell surface membrane, receptors on target cells. And then you could have given me anything more: activation of secondary messenger or enzyme cascade or signal transduction. Any of those points would have also got you a mark if you've written that. And question number 2, table 2.1 shows descriptions of 3 types of white blood cells. Complete table 2.1 stating the names of these 3 types of white blood cells. A large cell that has bean-shaped, kidney-shaped nucleus, can develop into a macrophage. That's a monocyte. Monocyte, I've always remembered it as a kidney-shaped as a monocyte. A cell that has a large spherical nucleus and little cytoplasm and it responds to non-self antigens, lymphocyte. You did not have to write anything B or T lymphocyte or just lymphocyte. It just said name. And then a cell that has a lobed nucleus and it is phagocytic. So that would be a neutrophil. You can't say phagocyte, but that's the part in the question. Neutrophil. So that got you your 3 marks. Then coming to the B part of the question, they have given you some sort of a comparison. Both are the same magnification, figure 2.1, 2.2. Dromedary camels are classified in the family Camelidae and live in desert habitats of North Africa and Asia. In these hot, dry environments, dromedary camels can lose up to 30% of their body mass from dehydration, causing their blood to become more viscous. So less fluid in it, it becomes very thick. Figure 2.1 shows a drawing of red blood cells of a camel. So this is the one of the camel. Let's write it on here. And this is a drawing of a human red blood cell. Well, we know that. So now looking at it, if you look at it, what is the difference? Now these are smaller, these are elliptical, those are round, and these are they are fewer, they are more. So let's look at the question now. Looking at it, you can see, the number in this space is this number of cells. Now, what are they asking you? They're asking you, they're saying figure 2.1 and figure 2.2 show differences between the red blood cells of camels and the red blood cells of humans. Suggest how these differences adapt camels for living in hot, dry. Now, a suggest I always say is a very difficult question. And so in a suggest here, you would just have to come up with something which is suggest how these differences adapt. So there are 3 marks. So allows easier or quicker flow of blood compared to human red blood cells. Why? Because they are smaller and smoother. They're not biconvex. Plus, you can give me any 2 explanations. Shape of the camel red blood cells is elliptical or lozenge shaped as compared to the biconcave disc of human cells. Camel cells are smaller than the human cells and a larger number of camel cells per unit volume. So the smaller they are will probably allow easier movement of blood. You see, humans are never does the blood become very concentrated. They've given you a situation in which in the camel they've showed you that it becomes very, very concentrated. So it shouldn't sort of become so concentrated it becomes semi-solid and is not doesn't flow in the blood vessels. So you have to think of it, what would be the possible 3 marks for it? So the first point had to be there, and then for the explanations, even if you gave me a whole lot of them, I could only give you 2 marks. So suggest how these differences adapt for living in hot, dry climates. So allows easier movement of blood, one mark, and the explanations were because they are lozenge shaped and why those are biconcave discs, and the camel red blood cells are smaller, so they will be easy to sort of flow in the blood vessels, and larger number of camel red blood cells per unit volume. So the transport of oxygen is more efficient because these are smaller than the human red blood cells. The llama is also classified in the family Camelidae. Llamas live in mountainous areas of South America, often at altitudes of 3,500 meters or higher. As the altitude above sea level increases, the air pressure decreases. The partial pressure of oxygen in the lungs of mammals at 3,500 meters is 6.4 kilopascals. Figure 2.3 shows the oxygen dissociation curve of adult human hemoglobin and adult llama hemoglobin. So we've got the adult llama hemoglobin, which is this one, and we've got the adult human hemoglobin, which is this one. So this is the human hemoglobin and this is the llama hemoglobin. Now you can see then, how do you see it? You see at any point, just see their oxygen, their saturation. Let's see it at 4. The human is nearly 58 or something like this. At 4 kilopascals, this is nearly 70. So one of them has a greater affinity. The llama one has a greater affinity for oxygen, doesn't give up its oxygen that easily. More affinity means it will be more percentage saturation of hemoglobin with oxygen. Any one point, just look at it, and that gives you a very good clue of it, what they're going to ask you in this question. With reference to figure 2.3, explain how the differences between the oxygen dissociation curves for human and llama show that llamas are better adapted for living at high altitudes than humans. These sort of questions have come in previous papers a lot. So at high altitude, hemoglobin of llama is more highly saturated with oxygen than hemoglobin of human. At 6.4 kPa, oxygen saturation is 86% for llama and 79% for humans. So it's nearly 6.7% higher. Llama hemoglobin has a higher affinity for oxygen than human hemoglobin. Llama hemoglobin has a higher affinity for oxygen than human hemoglobin. So more oxygen is transported or delivered to the tissues. So there'll always be sufficient oxygen unloaded in tissues to satisfy the demand. So with reference to it, we gave all the references and we're talking about that. Hemoglobin of llama more highly saturated, 6.4 kilopascals, 86% llama, 80% human. So there's about 6 to 7% higher oxygen saturation for llama. Llama hemoglobin has a higher affinity for oxygen, more oxygen delivered to the tissues, so sufficient oxygen unloaded in tissues to satisfy the demand. Then it says sketch a curve on figure 2.3 to show the effect of an increased carbon dioxide concentration on the percentage saturation of adult human hemoglobin with oxygen. Now this is of course the Bohr shift. So you'll draw a curve to the right of the human dissociation curve. So in this graph, all you had to do was draw a curve which was to the right. So you just have to make it a little more to the right, and then slowly it should reach there. Then explain the importance of the Bohr shift in metabolically active organs such as the liver. Now it's very easy. What will happen in the metabolically active organs release more carbon dioxide. So the partial pressure of carbon dioxide is higher. Now this decreases the affinity of hemoglobin for oxygen. So hemoglobin gives up its oxygen, and you know the formation of hemoglobinic acid. You see, you have to remember the carbon dioxide, water, carbonic acid, and then the hydrogen ions. So more carbon dioxide means more carbonic acid means more hydrogen ions. So hemoglobin tends to be friendly with the hydrogen and then decides to ditch the oxygen. So when it ditches the oxygen, that means it's decreased affinity of hemoglobin for oxygen. So more oxygen is released. Why? Because you see, if there's more carbon dioxide, that means there's more aerobic respiration. So we want to delay anaerobic respiration. So more oxygen is released for more aerobic respiration and more ATP production. So more ATP production is the most important thing per unit time. So release more CO2 as metabolically active organs will release more CO2, decreased affinity of hemoglobin for oxygen, and more oxygen released, more aerobic respiration, more ATP production. Question number 3, figure 3.1 is a photomicrograph showing part of a transverse section to the root Root of an iris, Iris jamanica. Irises are herbaceous monocots. These plants have the same transport tissues as herbaceous dicots, but the transport tissues are distributed differently in monocots. The central tissue in the root is parenchyma, which is the packing tissue. Now they've labeled that for you. They've shown you a root, and they've shown you something like R, S, and T. Now, cells R, S, and T are found in different tissues. Name the tissues in which the cells labeled R, S, and T are found. Now, R is the endodermis, S is the xylem, and T is the phloem. You know, these large vessels are always the xylem. These huge things like this is always the xylem. Remember that. This is always the xylem. So R is the endodermis, which is this part. And then S is the xylem. And then T is the phloem. So that was not very difficult. Name the tissues in which these are found. Now coming to part 2, outline the role of the tissues in which cell R is found. R is the endodermis. So what happens in the endodermis? To stop water moving through the apoplast pathway, or you can say to force the water movement to the symplast pathway, and so that water moves from the cell wall to the cytoplasm. And then this allows the control of substances into the root, or you could have talked of the Casparian strip. Then part 3, state an example of an organic compound that is translocated in the root of an iris. Very simple: sucrose, amino acid, protein, or you could say a named plant hormone. The electron micrograph shows a section through the same root cell in an onion allium cepa, and is magnified 6250 times. Draw a label line labeled with the letter P. So this would be a plasmodesmata. Any of this would be plasmodesmata. Label it with a P to identify one plasmodesmata. Table 3.1 contains information about 4 polysaccharides found in animals or plants. Complete table 3.1 by filling the missing information. Amylose, that's alpha glucose. Then cellulose is 1,4. Then glycogen is alpha glucose 1,4,1,6. It's an energy storage in animals. So that will finish the video here, and we'll continue the other 3 questions in the next video. Thank you very much.